Assalamu alaikum class. Today we are going to learn about chapter number 16 inherited chain. This is our second lecture. So this is the genetics. We are going to learn about the genetics right now. Now what is the genetics? It is the study of inheritance. And what is inheritance? This is the study of the transmission of characteristics or the traits from the parents towards the offspring. So over here, we are going to learn some terms which we have done in the previous class. These terms are the transcription and translation. So this is a kind of review of your previous knowledge. Actually, inside the cell, there is the nucleus, and inside the nucleus there is the DNA, which is of course double helix. This DNA carries the genes, which are the unit of inheritance, and these genes or DNA, it carries the whole information of that typical organism, living organism. So when the information is copied from the DNA towards the messenger RNA inside the nucleus, this process is called the transcription. So this is actually transcription. So messenger RNA, which is single-stranded structure, it has copied all the information from the DNA. So this messenger RNA will move out via nuclear pores into the cytoplasm. So this whole process is called the transcription, and now the messenger RNA moves towards the cytoplasm. This single-stranded messenger RNA has all the information, and according to this information, it has you have done the terms. For example, these are the codon. That is the triplet code. That is the triplet code. So there is the arrangement of codons over there, and there is a starting codon, and there is the stop codon over there. From the starting codon, the information will be copied in the form that the ribosomes, which have two subunits, larger subunit and smaller subunits, they get arranged themselves like a zip on the messenger RNA, and these ribosomal subunits will move forward in the form of the zip on this whole messenger RNA strand. And these ribosomes are going to actually read the information in the form of the codons on the messenger RNA, and then the transfer RNA, which has the anticodon at its base, anticodon which are actually anti to the codon. So these carries the anticodon. So the right anticodon will click the right codon, and these transfer RNA are carrying the specific amino acids with them, and then they will put the amino acid according to the correct information that is decoded by the messenger RNA from the DNA, and ribosome is going to read the instructions, and so it will form the polypeptide. Each amino acid will join and another amino acid will join by forming a peptide bonding. So polypeptide is formed at the end, and which get released when there is a stop codon at the end of the messenger RNA. So at the end, the ribosomal subunits will also leave the messenger RNA. The direction is always from 5 prime to 3 prime. You have also done this in your previous chapter, the chapter number 6, that the direction is 5 prime to 3 prime. So this whole process with the help of the messenger RNA and the transfer RNA and the ribosomal RNA, this whole process of making the polypeptide or the protein in turns is called the translation. So actually when it is a single polypeptide and its folding manner give it a typical protein structure, and all the bonds are involved. For example, it may contain the disulfide linkage, it may contain the ionic bonding, it may contain the hydrophobic interactions, it may contain the hydrogen bonding. So when all these bondings are involved, it will form a typical structure of protein. So this is called a translation, and this is actually one gene which is responsible for shifting the whole information, and the result is the protein synthesis. We call it the one gene, one polypeptide, or one gene, one protein, or one gene, one enzyme hypothesis. Actually, one gene is responsible for making the protein structure or actually enzymes are also the protein. So we can say that enzymes are also made by the same process. So this is the formation of protein, the transcription and translation process. The next one is the hemoglobin. The what is the hemoglobin? We are going to discuss with reference to the genetics, and we have done some terms on that day, and which we have done that what is a gene, what is the homologous chromosome, what is locus, what is centromere, and what is the allele, genotype, phenotype, homozygous, heterozygous, dominant, recessive, and we have done all these terms on that day in our class. And this is the hemoglobin. Hemoglobin is the oxygen carrying protein in the blood. There is another protein which is the myoglobin that is present inside the muscles. And actually, the hemoglobin, it contains the four polypeptides, two alpha and two beta, whereas the myoglobin, it contains a single polypeptide in it. So these four polypeptides each contain one heme group attached with it, and each heme group is responsible for binding the oxygen with its heme. So two alpha and two beta are there, and so the sickle, we are going to discuss with reference to the hemoglobin, the sickle cell anemia. Hemoglobin is present inside the RBCs, and due to the typical shape of the hemoglobin, that is of course globular protein, responsible for the functioning purpose. So this hemoglobin, it gives an oval shape to the RBC, and due to this oval shape, the RBCs are actually responsible for the transport of oxygen throughout the body, because of this oval shape, the RBCs can squeeze out, squeeze from the capillaries easily. So actually, when there is the normal hemoglobin is present with no mutation, no malfunctioning, then this is the oval shape RBCs are present. But if there is a genetic disorder, that is the sickle cell anemia, there is actually a one beta chain mutation. Actually, one beta chain mutation is there. Due to that mutation, the beta chain structure distorted, and as a whole, only a single amino acid is replaced by another one, glutamic acid by the valine amino acid. So, due to a single mutation in one beta chain only, there is a kind of mutation over there, and this mutation is actually recessive in nature due to the recessive alleles. So the recessive alleles, it takes place. Recessive means that it occurs only in by the if it is homozygous recessive. So we can say that we can denote HbA for normal, and that is for normal beta globin polypeptide, and HbS for sickle cell beta globin polypeptide due to a mutation. So this is a disorder. The sickle cell is disorder. So if the condition is HbA and HbA, it means that is normal. And if it is the HbS or HbS, that is the sickle cell. And both these conditions are the homozygous condition. So this disease will appear only if it is homozygous recessive, because this is due to the recessive alleles. So it means that HbA is dominant over the HbS of the sickle cell anemia. Actually, when we talk about the sickle cell anemia, the normal shape of the RBCs, which was oval, they will turn into the sickle cell, and due to this, there is a problem in the transport of the RBC throughout the body, and especially when they are moving from the narrow capillaries, and due to the stoppage of these RBCs at certain points, there will be no further transport of oxygen in the body of that person. So we call that condition as a sickle cell anemia. Yes. राय उमर।
मिस ये जो हेट्रोजाइगस कंडीशन है,
जी
मिस इसके अंदर भी तो मिस इसके अंदर सारे के सारे ब्लड सेल्स जो है वो नॉर्मल होते हैं, या कुछ सिकल सेल भी, कुछ सिकल शेप भी होते हैं?
but I'm just going toward that condition, that is the heterozygous condition. I'm going to discuss that condition. First of all, I was discussing about the homozygous condition, both in the normal and the sickle cell anemia. Now, the third condition is the HbA and HbS, which you are talking about that, Rayuman. Actually, when we talk about the HbA or HbS, it means that is the heterozygous condition. Heterozygous condition is actually uh, in case of the heterozygous condition, uh, that is mixed condition. Some cells are normal and some cells are sickle shaped, but normally they behave just like the normal people. We call them the carrier, or we call the whole, we call this one this one sickle cell anemia. Anemia is the shortage of the iron, right? We call this the sickle cell anemia, but we call this condition, the heterozygous condition, the sickle cell trait. It means trait, they are having the trait, they are the carrier, they are the carrier, they are having the trait. They may have the mixed type of blood cells in them, RBCs in them. Some are sickle shaped, some are normal. They are heterozygous, they are the carrier. It means they can transmit their genes or alleles in the next generation in the only in the homozygous recessive condition. Now what happens? Normally they people behave very normal, just like us, but it is difficult to judge them if there is no kind of test of them. For example, they have no breathing problem, no breathing issues, but at certain conditions, if for example if they are moving towards the high altitudes, or if they are running very fastly, or if there is a kind of any problem, if there is a kind of any shortage of oxygen at certain places at certain times. So in you can say the ultimate conditions, they may suffer the symptoms of the sickle cell anemia, but normally they behave like the normal people, right? So yes, Shazir.
मैम जो हेड्रोसाइगस कंडीशन है मैम,
Hmm.
मैम इसमें HbA डोमिनेंट होगा और
बिल्कुल
HbS
बिल्कुल
रिसेसिव होगा?
बिल्कुल HbA डोमिनेंट है, तभी तो देखे ना, मैंने बिल्कुल स्टार्ट में बात की दैट HbS is the sickle cell beta globin. It means that the sickle cell is actually the recessive condition. I talk about that this is due to the recessive allele. तो हमेशा जो डोमिनेंट होता है वो रिसेसिव को सप्रेस कर लेता है। लेकिन अगर कंडीशंस ऐसी हो जाएं कि कहीं बहुत ही शॉर्टेज हो जाए ऑक्सीजन की, कहीं वो ऊंची जगह पे चले जाते हैं, कहीं पे हाई एल्टीट्यूड पे, तो उनको प्रॉब्लम हो सकता है। जहाँ पे शॉर्टेज ऑफ ऑक्सीजन ऑलरेडी है तो उनको प्रॉब्लम हो सकता है। अदरवाइज इनका नॉर्मली नहीं पता लगेगा जब तक आप इनका टेस्ट नहीं करवाते कि इनका जीनोटाइप क्या था। तो ये बहुत रेयर कंडीशंस में
जी
ये बिहेव कर सकते हैं, बिकॉज दिस इज नॉट द दे आर नॉट द अनीमिक पीपल, they are actually having the trait, they're actually having, they're actually the carrier, right? So this is the condition, right? So clear? Okay.
जी मैम जी।
Okay, the next thing is that we're going to make the cross of these two you can say the genotypes. For example, if there are two parents, yes, Rai Umar.
मिस ये जो सिकल सेल एनीमिया वाले लोग होते हैं मिस, इनमें क्या नॉर्मल ब्लड सेल्स होते ही नहीं है?
बेटा नॉर्मल भी होते हैं, मिक्स होते हैं। नॉर्मल ब्लड सेल्स होते हैं, बिल्कुल होते हैं, और बहुत कम क्वांटिटी में जो उनसे हैं वो सिकल भी होते हैं। बहुत कम क्वांटिटी में
अ,
सिकल भी हो सकते हैं। जी
नहीं मिस, ये जिनको जिनको सिकल सेल एनीमिया है, मतलब
जी
जो
जी।
होमोजाइगस रिसेसिव है,
जिनको सिकल सेल एनीमिया
मिस
है,
उनमें
अच्छा अच्छा, उनकी बात कर रहे हैं। नॉर्मली उनमें अगर होंगे ना नॉर्मल सेल्स, तो वो क्योंकि उनका तो देखिए उनके तो जेनेटिक डिसऑर्डर आ गया है। अगर नॉर्मल सेल्स हुए तो बहुत, आई डोंट थिंक सो कि उनमें नॉर्मल होंगे, पहली बात तो ये। और अगर कोई ऐसी रेयर कंडीशन आती है तो इतनी कम क्वांटिटी में होंगे, इसका मतलब ये कि उनके तो नॉर्मल सेल्स एज सच नहीं होंगे। और जो उनसे हैं सिकल शेप होंगे। सिकल शेप होने की वजह से वो जब कैपिलरीज में से पास नहीं करेंगे तो फिर इशू आएगा, और फिर उनका होगा कि उनका ब्लड जौन सा है वो उनका करवाना पड़ेगा, चेंज करवाना पड़ेगा। लेकिन अगर उनकी स्वेयर कंडीशन है और अगर इतने एक्सपेंसेस जौन से हैं वो कर सकते हैं, तो फिर जाहिर सी बात है कि उनको वो हम उनको कहेंगे कि वो जीन थेरेपी जौन सी है करवा सकते, बोन मैरो ट्रांसप्लांट करवा सकते हैं। लेकिन आरबीसीज मिक्स्ड हो सकते हैं, लेकिन उनमें प्रपोर्शन जो सी है वो रेस कर सकती है सिकल सेल की। जी
ओके मिस।
यस शाहजेब।
जी मैम वो सवाल का जवाब मिल गया मैम।
ओके बेटा,
सॉरी।
it's okay. Okay, now we have two kind of parents over there, and both are the heterozygous condition, that is HB and HBS, both are there. And we are going to make a cross by putting them in the Punnett scale. From HB, this is the parent 1, this is the parent 2. Of course, one is mother and the father. So we are when these are denoted by the gametes. This circle denotes the gametes. So HBA and HBA will make the HBA normal. HBA, HBS, this one trait or carrier. HBS, HBA, trait, HBS, HBS, sickle. So it means one is normal, two are carrier, and one is sickle cell anemic person. The genotype means the combination of the genes. So the genotype is 1, 2, 1, right? And the phenotype is 3 behave like normal, right? And one is specifically sickle celled. So it means that ratio is 3:1. The ratio is phenotype is 3:1. So this is a kind of you can genetic diagram of if you people are asked to make a genetic diagram, how to make a genetic diagram, inshallah we will discuss later on very clearly in next sessions. Okay.
मैम जो सिकल
जी जी
सेल की मैम रेशो है मैम,
जी जी
अगर वो कम होगी तो मैम तो नुकसान तो नहीं होगा ना कि
नहीं तो हाँ उसमें बेटा मलेरिया वाला इशू जो है
मैं
वो
आपसे
डिस्कस।
पूछूं कि नुकसान
हाँ
तो
जी,
नहीं होगा कोई?
नहीं। अच्छा ये जो लोग हैं ना अफ्रीका में, उनमें जिनमें सिकल सेल अनीमिया होता है तो वहाँ पे मलेरिया उन लोगों को नहीं होता, क्योंकि जब ब्लड सक करते हैं जो मॉस्किटो होते हैं तो वो उस ब्लड को सक नहीं करते हैं प्रॉपरली, जिसमें जो सा उसको इन्फेक्शन नहीं होता है, जिसमें जो सा है सिकल शेप्ड आरबीसीज होते हैं। तो जो लोग हेट्रोजाइगस कंडीशन में हैं तो ज़्यादातर जो होमोजाइगस रिसेसिव हैं जो कि सिकल सेल अनीमिक पीपल हैं, वो उनको तो बिल्कुल ही नहीं होगा मलेरिया, बेचारे तो एक बीमारी से लड़ रहे हैं। और जो दूसरे जो हेट्रोजाइगस कंडीशन में हैं उनको भी वो बच सकते हैं मलेरिया से, क्योंकि उनकी आरबीसीज की जो शेप जो सी है वो नॉर्मल नहीं है, राइट? ठीक है, इनको एडवांटेज है।
जी मैम।
ठीक है। Now the next topic is about the multiple alleles. So far we have discussed that a genes carries the two alternative form. These are called the alleles. But actually, the genes may carry more than two alleles, right? The example is the blood group system, and there is I gene antigen on the 9th chromosome that is responsible for for marking the blood group to that person. 9th chromosome, that of course that is the autosomes, because we have discussed that x-axis for female, xy is for male, and these are the sex chromosomes. And this is the 23rd pair of the chromosome, and other this 23rd pair, the rest one are the autosome, they are not the sex chromosomes. So this I gene which is present जी जी
मैम जो ऑटोसोम में होंगे ये मैम होमोलोगस पेयर होंगे इनके?
जी होमोलोगस होंगे, क्योंकि जो सेक्स क्रोमोसोम है वो इस तरह से भी हो सकते हैं, बोथ आर एक्स, इट मींस दिस इज द फीमेल, the male has X or Y, both have different shapes. So the autosomes, they may behave like the homologous pairs, right? So of course, one comes from
जी
male and one comes from the female, or one comes from father or mother. This is the condition, right? So on ninth chromosome, gene I is present. Instead of having two alleles only, this has three alleles. I gene on ninth chromosome has three alleles: A, B, and small I or I naught. So if the condition is in the home in this this is actually representing if that is a kind of you can say the haploid condition. If the diploid condition is there, for example, IA and IB, it's denoting the diploid condition. If it's half, it means it is showing the gamete only. This is diploid condition. If this condition is present, and if the heterozygous condition IAI is present, it means the blood group A is present. Of course, A is completely dominant over the small I or I naught. So it means that it is showing the complete dominance. This condition is called the complete dominance, right? And if it is IB, and IB small I, then of course again B is dominant over I, but in heterozygous condition, so it means that is the blood group B. And if it is IAIB, that is called the codominance. No allele will suppress the effect of other. Both are equally contributing towards the, you can say the phenotype or genotype. This is called the codominance. If both alleles are coexisting and they are both are showing their effect simultaneously, this is called the AB. In case of the blood group system, this is called the codominance. And if it is I naught, then this is the blood group O. In the homozygous recessive condition, the blood group O. Okay. Now, when we call about the sex inheritance, that is the, of course, which is responsible, which is affiliated with the sex chromosome XX in female and XY in male. So this is about the sex inheritance. Now the next one is the incomplete dominance. We have talked about the complete dominance and we have discussed the example of the complete dominance in our class also. I have given you the example of the capital T and small t in case of the pea plants. That is, the capital T is responsible for the tallness of the plant, and the small t is responsible for the shortness of the plant. And we have made that cross, and how to make the cross. First of all, you need to label the P1, and then the gametes one, then the crosses, then the first filial generation F1, then the P2, then the G2 gametes 2, then the F2 second filial generation. These terms are very important in this regard. So when we talk about the incomplete dominance, for example, there is a capital R allele for red flower, small r for white flower, and we are going to make the cross. This capital R, small r are both are homozygous. We call them the pure breed or true breeders, and this is the P1, and we will make the gametes G1 like this. So all the F1 generation will be capital R, small r. We expect that capital R is dominant over the small r. We expect that
Miss,
yes, जी
मिस ये ऑल पिंक क्यों होगा, रेड क्यों नहीं होगा?
यही बताने लगी हूँ, ठीक है. So we expect that all should be red, but the results are so different. All were pink. Why? Because in this case there is no dominance of one allele over the other. There is a kind of mixing or blending of the two alleles in the form of the phenotype. The genotype remain the same, वो तो वही रहेगी, heterozygous ही थी, लेकिन जो हमें phenotype नजर आई वो blending की तरफ चली गई. बजाय ये कि चाहे जैसे हमने dominance में पढ़ा कि एक जीन ने दूसरे को suppress कर दिया, वो भी नहीं हुआ, और फिर हमने पढ़ा co-dominance में दोनों genes alleles मिलके रह रहे थे, वो भी नहीं हुआ, but both alleles form a kind of blender or mixture. that results in the phenotype all pink. This condition is called the incomplete dominance, which is a type of the dominance, incomplete dominance. It means that actually this is another form of dominance. What is this one? This is the first form is complete dominance, the next form is codominance, and the next form is incomplete dominance. The other forms also, but over here we are going to discuss about the forms, the three forms incomplete dominance. So ये हम इसको discuss करेंगे with reference to it, inshallah, with reference to the मेंडल लॉ ऑफ इनहेरिटेंस, उसके रेफरेंस से डिस्कस करेंगे। तो ये किस तरह से रूल्स को फुलफिल करता है। बट if we make a further cross of this r, capital R small r with another capital R small r, the results will be same 1:2:1 जो हमने बाकियों में पढ़ा था। लेकिन वो क्या होंगे, उसमें से कुछ रेड होंगे, कुछ व्हाइट होंगे, और जो हेटेरोजाइगस कंडीशन में होंगे दे विल बी ऑलवेज पिंक। तो लेकिन रेशियो जो सी है फिनोटाइप डिफरेंट आएगी, लेकिन जो जीनोटाइप जो सी है वो वही आएगी जो हम पहले जो पढ़ते आए हैं, 1:2:1 आएगी। ठीक है। सो फॉर एग्जाम्पल इफ वी डू नॉट हैव द जीनोटाइप ऑफ एनी क्रॉस, फॉर एग्जाम्पल हमारे पास टी है टॉल के लिए और स्मॉल टी है शॉर्ट के लिए, हमने ये देखे इनके गैमेट्स बनाए, ये हमने गैमेट्स बनाए, दीज आर द पेरेंट्स वन, दीज आर द गैमेट्स। और फिर हमने क्या किया कि ये ऑल टॉल आए, हमने इनका जो थे ना ये हेटेरोजाइगस थे, इनका क्रॉस अब हमें नहीं पता था, ये जो सारे टॉल आए हैं ना हमारे पास, वी डोंट नो कि हमें इसकी फिनोटाइप तो नजर आ गई थी दैट ऑल वर टॉल, लेकिन हमें ये नहीं पता था कि ये होमोजाइगस वाले टॉल हैं या हेटेरोजाइगस वाले टॉल हैं, हमें इनकी जीनोटाइप का नहीं पता था। सो हाउ कैन वी फाइंड द जीनोटाइप ऑफ दिस कैपिटल टी एंड स्मॉल टी? हाउ? अगर हमने इसकी जीनोटाइप निकालनी है तो हम ये ले लेंगे, हमें नहीं पता कि इसकी जीनोटाइप क्या है, हमें सब टॉल दिख रहे हैं, हमें नहीं पता ये होमोजाइगस है, हमें नहीं पता ये हेटेरोजाइगस है। हम इसको क्या करेंगे, हम इसका प्योर ब्रीड लेंगे रिसेसिव वाला, ये खुद से लिया हमने, स्मॉल टी होमोजाइगस रिसेसिव, इसको लेके उसके साथ जो हमें नहीं पता था अननोन जीनोटाइप वाला उसके साथ क्रॉस करवा देंगे। क्रॉस करवाया तो क्या आया, क्रॉस करवाया तो आया 50% टॉल हेटेरोजाइगस थे, लेकिन हमें भी पता जीनोटाइप उसकी क्या है, और 50% शॉर्ट। अगर ये सारे के सारे कैपिटल टी होते तो फिर तो ये ऑल टॉल होते जैसे कि हमने ये इसमें पढ़ा था, लेकिन अगर ये 50% टॉल आ गए और 50% शॉर्ट आ गए, इसका मतलब ये जो हमारे पास अननोन जीनोटाइप क्या थी, वो हमारे पास थी हेटेरोजाइगस थी। दैट्स व्हाई ये टॉल भी आए और शॉर्ट भी आए, अदरवाइज तो ये ऑल टॉल होते अगर ये होमोजाइगस होती, जिस कंडीशन इन विच वी क्रॉस द टू नो द genotype of the parent, we cross them with the homozygous recessive to know the genotype of that parent, that is called the test cross। आपने जो भी रिजल्ट आया आपको नहीं पता, ये अननोन था, आपने इसको क्रॉस करवा दिया उसके होमोजाइगस रिसेसिव के साथ खुद से। अगर तो ऑल टॉल आते तो ये क्या होता, जो उसका पेरेंट होता ये वाला, ये होता होमोजाइगस, और अगर ये 50-50 आए उसका मतलब है ये किसकी जीनोटाइप क्या है जी, ये हेटेरोजाइगस है। दिस इज द वे टू फाइंड द जीनोटाइप ऑफ द पेरेंट्स, this term is called the test cross। so
मिस
आप जी
मिस जब ये ऑल टॉल आ रहा था तो तब भी तो हमें जीनोटाइप का पता चल रहा था कि होमोजाइगस है।
रिकॉर्डिंग ऑफ करते हैं जी बेटा, जी आप बोले दोबारा
मिस
बोलें।
मैं कह रही हूँ जब ये ऑल